Okay. So within among the bacteria itself, they have different strategies. We call it life strategies. Okay. So there's a R strategy and a K strategy. This is not only restricted to microorganisms, but actually this is from the science of ecology, where they realize they can categorize organisms as either having R strategy or K strategy. So R strategy, an organism that adopts R strategy are usually copioptrophs with high reproductive rates but little competitive adaptation. So these are organisms that grow very fast when there's a sudden ample supply of food. So they respond fastest to a sudden input of food. So they'll grow very fast. But they also have very little competitive adaptation, which means once the food is no longer available, they will lose out to the uh, other competitors. Okay. So for in the microbial world, bacteria that are known to adopt R strategies include Pseudomonads, Bacillus, Cyanobacteria, and Aspergillus. Whereas for the other K strategy, they are mostly oligotrophs with low reproductive rates but with diverse competitive adaptation. So these are bacteria that do not respond as fast to a sudden nutrient input, but they are able to maintain themselves even when the food supply is low. Do you all hear me?
I can.
Yes, doctor.
Oh,
I
you can hear.
can. I can hear as
Oh,
well.
you can't hear me. Can you hear me now?
Yes, doctor, I can hear you now.
Okay. Okay, let's continue. So, I was talking about R and K strategy. So, K strategy organisms are usually oligotrophs with low reproductive rates. So, they might not respond so fast to a sudden nutrient input, but because they have diverse competitive adaptation, they are able to maintain their population even when the nutrient supply is low. They are able to compete better, adapt to different sources of food. Okay, for example, your soil streptomycetes, spirula, marine vibrios, agrobacterium, and corynebacterium. So you have organisms that adopt R strategy and also organisms that adopt K strategy. So when you look at their definition, you realize that in certain conditions, like for example in waters where there is a lot of nutrients, you realize that most probably the R strategies will grow better here because the nutrient level is high. Okay, so the copioptrophs will be able to grow better. and will be able to predominate in that kind of environment. Whereas for waters with low nutrient conditions, you will probably assume that the most of the microbes there are oligotrophs, okay, adopting K strategy lifestyles because they are able to adapt better to low nutrient levels. So if from our own work, we found that even within the same genus, okay, we were studying Vibrio species. Even within the same genus itself, we found that some Vibrio species adopt R strategy, some Vibrio species adopt K strategy. What was interesting is it was the pathogenic Vibrio that adopted the K strategy. Okay, it was the pathogenic Vibrio, for example, Vibrio parahemolyticus, Vibrio vulnificus, that adopted K strategy lifestyles. Okay. So that would also explain why we don't see so many pathogenic Vibrio in the seas. Why we don't often isolate them, because they are also adopting different life strategies. Okay, but because they maintain K strategy, they are able to maintain their population even though uh, their counts are very low. Okay, so this is the difference between uh, microbes adopting R strategy and K strategy lifestyles. Okay, so when you have microbe and microbe interaction, when they live together, so they might be adopting different lifestyles, but there is also sometimes interaction between them, okay, between the different microbial populations. So similar to classical ecology, we can also find examples of commensalism, symbiosis, parasitism in the microbial world itself. Okay, for example, commensalism is when one population benefits without any benefit or detriment to itself. Okay, so uh, commensalism comes from mensa, which is a Latin word for table. So it's more, think of it as eating off the table scraps. So, for example, in a cafeteria, okay, for example, sometimes in the university cafeteria, you see cats eating off your table scraps. So that could also be a form of commensalism, where the cat is eating off your uh, table scraps. So the cat benefits, but it doesn't uh, do anything to you. It doesn't bring you any damn benefit or detriment to yourself. Okay, so that's a form of commensalism. So this is a common but non-obligatory relationship. Non-obligatory means the cat can eat the table scraps from another person. Okay, there is no obligation for the same cat to be eating off your table scraps every time. Okay, so that's the definition for commensalism. So in the microbial world, uh, in a water cooling system, aquatic habitat, the Flavobacterium brevis excretes an amino acid that is required for growth by Legionella pneumophila. Okay, that's why Legionella pneumophila can persist in water cooling systems, and sometimes they cause uh, legionellosis in air conditioning systems. So this is a form of an example of commensalism, because whether Legionella pneumophila is present in the water cooling system, the Flavobacterium brevis will still excrete the amino acid because it is a byproduct of its physiological process. So it excretes the cysteine, the amino acid, whether or not uh, Legionella pneumophila is present. Okay, so this is a form of commensalism. Legionella pneumophila will benefit without any harm or any benefit to Flavobacterium brevis. Okay, so the next is synergism. So this is where both the population benefits. So in commensalism, only one population benefit. In synergism, both populations benefit. So the difference between synergism and mutualism or symbiosis, is whether it's obligatory. In mutualism, the relationship is obligatory, whereas in synergism, it is non-obligatory. So non-obligatory is the same as the earlier example, which means there is no requirement for the two populations to be uh, living together, to be interacting. Okay, there's no requirement at all. However, when they are together, they benefit each other. Okay, so a good example from the microbial world will be chlorobium, which carries out carbon dioxide fixation and sulfide oxidation to form formate and sulfur. So this is using light, so it has to be in the upper layer of the, for example, lake system. Okay, then the formate and sulfur that is produced is used instead by spirillum. Okay, so formate as a carbon source and sulfur as an electron acceptor. And when they utilizes formate and sulfur, spirillum releases carbon dioxide and H2S. So these two are instead the substrate for chlorobium. So both of them benefit each other. The product from one organism, is used by the other. The product from each other is used by the other party. So both supply each other's nutritional needs. So there is synergism. Okay, so this is a good kind of mutually benefiting relationship. So for mutualism, another word is symbiosis. Both partners also benefit. However, the interaction is obligatory, which means the partners cannot be replaced. So the relationship is highly specific, and this kind of relationships usually have evolved over a long time. So when this kind of relationships have evolved over a long time, they no longer can be replaced by other microbes. Okay. A good example will be lichen, okay, where both cyanobacteria and fungi enters a symbiotic relationship. Okay, so the what happens is The cyanobacteria in lichen carries out photosynthesis and produces photosynthetic products, for example, carbon source. And cyanobacteria also fix nitrogen. So these products are used by fungi, utilized by fungi. Fungi is a heterotroph. What the fungi offers is it offers attachment to the substratum. Okay, so it and offers a habitat for the cyanobacteria to live in. Okay, the attachment by fungi with its mycelial network also increases absorption capability. So the fungi is able to take up water and nutrients from the substratum more efficiently than the cyanobacteria. So the fungi helps out uh, in the form of habitat, attachment, and water and nutrients from the substratum, whereas the cyanobacteria offers photosynthetic products and fixation products to the partner. So in this kind of relationship, both microbes benefit. So here is another kind of interaction, amensalism or antagonism. This is where uh, one population doesn't benefit, while the other population uh, is harmed by the other population. So one population is experiencing a negative effect. Is experiencing a negative effect from the interaction, whereas the other organism is neither benefiting or experiencing negative effect. So, however, when you look at this kind of definition, it is very hard to prove that the byproduct from one population that is inhibitory towards the other doesn't actually cause a gain for the other population. Okay, so what I mean is, the definition of amensalism says that one population is inhibited. One population is inhibited, or one population experiences detrimental effects, whereas the other does not gain. However, you know that in the microbial world, where if the microbes live close together, when one microbe is losing out, the other will definitely gain. Gain either from physical space, gain from food. Okay, in the other population will usually gain when one of the population is in detriment. So amensalism is very hard to prove. Okay, that's why it is harder to find uh, specific examples for amensalism. So the example here is very general, where one population, the life process of one population changes the environment that it is detrimental to another population. For example, the respiration by one microbial population, will reduce the oxygen level. So if one microbe is facultative anaerobe, the other microbe is aerobe. So the reduction of oxygen level will be detrimental to the aerobe. the facultative anaerobe will still be okay. But if you think about it, when the aerobic microorganisms die off, the facultative anaerobe will eventually gain. Okay, so it is hard to prove. That's why there's no specific specific example from the microbial world here. However, um, the definition of amensalism is like this. Okay, one population is in detriment, the other does not gain. Okay, any questions? So one last will be parasitism. Parasitism is more straightforward. One population benefits while the other is harmed. So parasitism, you have a parasite-host interaction. One population benefits, the other is harmed. Predation is also a form of parasitism when you think about it, okay? Because the predator gains, the prey loses, okay? However, predation is short-term, okay? Parasitism is generally can be a long-term interaction, either through endo or ectoparasites, and because it can be a long-term interaction, this kind of a parasite-host interaction is usually specific and have usually evolved over time. Okay, for example, specific bacteria fudge with specific bacteria. So these kind of parasite-host interactions are specific and have evolved over time. Okay, any questions? Okay, so so when you look at the microbial communities, okay, there are different levels of ecological organization that you can also adopt from the um, classical ecology. So an individual, one, a sum of individuals make up a population. Then when you have two different populations that share the same habitat, share the same niche, you have a guild. Okay, so when you see this, then if they share the same habitat, they share the same niche, they use the same resource, that means there is already some form of competition. Okay, so when you have different guilds, then you start uh, seeing a resemblance of a community. Okay, you, although there is competition among the within the guild itself, if different guilds uh, they might have the same habitat, they might live on the same habitat, but because they occupy different niches, maybe one is uh, maybe one prefers carbohydrates, one prefers amino acids, different substrates, then the level of competition is not so clear. Okay, so they are able to live together within the same habitat. Okay, then when you have lots of communities living together, then you generally have a ecosystem. So these are the different levels of ecological organization. Any questions before we move to the next topic?
No,
Okay,
doctor.
so we've talked about how the bacteria can uh, how bacteria have different life strategies. Okay, how their interaction, the different categories of interaction between them, whether mutualism, commensalism, antagonism, and so forth. Okay, and also uh, the different levels of ecological organization that's possible within the microbial world itself. So, but for the microbes to be interacting, for the microbes to have this kind of complexity in their organization, there has to be a form of communication. Okay, basically, imagine a microbe is so small. There is no way it can sense its surrounding. Okay, I mean it cannot, not like humans. We cannot. It doesn't visualize the surrounding. So, but they must have a way to know, uh, to sense its surrounding. It must have a way to know whether there are what is happening in its surrounding. So there is one form of communication that scientists have found that is being used by bacteria, and now we know that it is being used widely in bacteria, and that's called the quorum sensing. Okay, so quorum sensing is a form of cell-to-cell communication through the exchange of chemical signal molecules. So these chemical signal molecules are called autoinducers. So it's called quorum sensing because this kind of a system allows the bacteria to sense what is their number. Okay, so they know whether they are low in numbers or high in numbers. Okay, through this autoinducer. The most common autoinducer are the acyl homoserine lactones autoinducer. So they share a common uh, basic structure. Okay, a homoserine lactone. So quorum sensing research actually began in the early 1970s. Okay. At that time, a group of researchers were looking at the bioluminescence of Vibrio fischeri and Vibrio harveyi. Okay. So two different Vibrios that carries out vibrioluminescence. Uh, uh, they carry out bioluminescence. So when they grow this bacteria in the flask, chick flask method, they realize that the luminescence only occur in the late log phase. Okay, the late exponential phase. So only when the numbers reach a certain amount, only when the vibrios reach a certain number does the bacteria start to luminescence. They wanted to know what triggered the luminescence. So what they did was they took the filtrate. Okay, they took the filtrate from a late log culture. So they filter off all the bacteria, and they only took the filtrate. Then they place the filtrate into a fresh early log phase culture. and they found out that the bacteria luminesce immediately. Okay, that means something in the filtrate can trigger the luminescence response. Okay, so from there they isolated the molecules, and then they name it autoinducer. Then now we know more and more about this quorum sensing system. So the quorum sensing, the term quorum came about because the system senses the number of microbes that are present. So whether there's enough quorum or not for the bacteria to begin a specific response. From the experiment, they also found that the filtrate from fishery doesn't stimulate Hawaii, whereas the filtrate from Hawaii also doesn't stimulate Vibrio fishery. So there is specificity also in the quorum sensing system. Okay. So now researchers have found lots of different kinds of systems, quorum sensing systems in bacteria. A common one is the LuxIR circuit. Okay, LuxI is inducer, LuxR is the reporter. Okay, so this is common in the gram negative bacteria. Okay, so the LuxI gene will synthesize the autoinducer, an acyl homoserine lactone, and this acyl homoserine lactone diffuses freely out of the bacteria. Okay, so every bacteria will produce a specific amount of autoinducer. Okay. So if more bacteria is present, the concentration of AHL also increases. And once it reaches a threshold, the concentration of AHL, you'll activate the LuxR gene, which will start a targeted gene expression. So the gene expression here is dependent upon the concentration of AHL. So that is how this bacteria can sense the number of bacteria present through the concentration of AHL. Okay. Any questions? Okay. So there is another quorum sensing system circuit. It's called the oligopeptide 2-component circuit. This is more common for gram-positive bacteria, but it is also present in gram-negative bacteria. Okay, so for this, the difference is clear. It doesn't generate AHL, the autoinducer, immediately. Okay, instead it produces precursor peptides, and these precursor peptides do not diffuse freely also across the membrane. There's a dedicated transporter that brings out the precursor peptides. And as it transports the precursor peptides, it will also modify it to make it into a auto-inducing polypeptides. Okay, so each bacteria will produce their own auto-inducing polypeptide. And when the concentration of auto-inducing polypeptide is high enough, it will activate the histidine kinase on the cell wall, which will phosphorylate a response regulator and starts gene expression, targeted gene expression. Okay, so this is more common in gram-positive bacteria. Why quorum sensing is was so hot 10 to 5 years ago, 5 to 10 years ago, was because most of the virulence genes require some form of quorum sensing system to initiate the expression. Okay, so it created a lot of excitement in microbiology because if the pathogenicity of bacteria is controlled by quorum sensing systems, that means if we can control quorum sensing system, we can prevent bacterial virulence. Okay, because the problem of antibiotic resistance still exists. So scientists begin to think that maybe disrupting the quorum sensing system is one way of overcoming antibiotic resistance. Okay, one way of uh, preventing bacterial virulence. So that's why a lot of money was pumped into quorum sensing research. So in UM, I was in UM, Dr. Chan Kogan. So he's probably the top quorum sensing scientist in Malaysia. He has done a lot of work on quorum sensing. Okay, so there's a lot of publication you can see. So now we know that bacteria can have more than one quorum sensing system, which means it's not just one bacteria, one quorum sensing system. Okay? A bacteria can have two or more quorum sensing systems for different targeted gene expression. So it's not just one to one. It can have more than one. Earlier I mentioned how the filtrate from Vibrio hawaiii doesn't elicit luminescence in Vibrio fischeri. Okay, so there is some form of specificity also in a quorum sensing system. But now they also know that there is also interspecies cell-cell communication where several bacteria can recognize the same autoinducer. Okay, so the E. coli will know how many cholera, streptococcus, the number of microbial population including these two within the community. So there is also interspecies communication. Okay, so they found quorum sensing systems that target gene expression for bioluminescence, for secretion of virulence factors, for biofilm formation, sporulation, conjugation, and pigment production. So biofilm formation is also a part of can be a part of a bacterial pathogenicity. Okay, it can be also part of a bacterial virulence factor because biofilm will prevent the entry of antibiotics into the bacterial cell itself. So it is also a form of defense for the bacteria. That's why that a lot of it has become an exciting prospect if we are able to control so interfere, if you are able to interfere with these quorum sensing systems for these processes, then we can prevent virulence, prevent biofilm formation, prevent other processes that promotes bacterial virulence. And there are a few strategies, two main strategies that researchers are undertaking to interfere with quorum sensing system. One is by using an analog. Okay, it's structurally similar chemical, but it doesn't work. So what it does is it binds to the response regulator, but it doesn't elicit the targeted gene expression. So it interferes with the gene expression. For example, they found that a macroalgae produces a halogenated furanol that interferes with bacterial attachment on its surface. Okay, so they are able to use this analog to interfere with quorum sensing systems in bacteria. Or the use of quorum quenching systems. Some researchers have said if we can find bacteria that degrades the autoinducers, maybe then we can regulate or control quorum sensing systems.